Hey everyone! Today we are diving into the absolutely mind-blowing science that just snagged the 2024 Nobel Prize in Chemistry. It's a story about cracking one of biology's oldest, toughest puzzles and, get this, learning how to actually build the tiny machines of life from scratch. So let's jump right in. So where does this incredible story start? Well, you might be surprised to hear it doesn't really begin in a chemistry lab at all. It starts in the world of philosophy, with one of the key scientists behind this whole revolution, David Baker. And believe me, his path to a Nobel Prize was anything but ordinary. I mean, seriously, how great is that? You have a Nobel Prize winner in chemistry openly admitting that he didn't even really like the subject at first. It just goes to show you that sometimes pure, simple curiosity can take you down the most amazing and totally unexpected roads. Okay, so let's just trace this wild journey for a second. He's at Harvard, deep in philosophy and social sciences. Big picture questions about the world. But then a spark of interest in biology just changes everything. So he pivots hard. He heads over to the University of California to study biochemistry, and that is where he decides to take on one of the biggest, gnarliest challenges in all of science, the protein folding problem. So what exactly is this grand challenge, this protein folding problem? Well, to get it, we need to talk about this beautiful, super complex art form. Think of it like microscopic origami, and it's happening inside every single one of your cells, right this very second. You see, proteins are basically the little workhorses of life. They are the primary actors, the tiny machines that do almost everything. They're running chemical reactions, helping your cells divide, making it possible for you to move. I mean, you name it, a protein is probably involved. If you want to understand life, you have to understand proteins. And here is the absolute key. A protein starts as just a simple string of building blocks, these things called amino acids. But its job, its function, is all determined by the super intricate 3D shape it folds into. Its shape is its purpose. And these amino acids have different personalities. Some are hydrophobic, they hate water, while others are polar, they love it. So for decades, the billion-dollar question was, how can we possibly predict that final functional shape just by looking at the sequence of amino acids? Now, for the longest time, the only way to figure out a protein's 3D structure was through this incredibly difficult, painstaking lab work, like X-ray crystallography. These methods were painfully slow, crazy expensive, and honestly, for a lot of really important proteins that were too big or too wiggly, they just didn't work at all. The absolute dream was to build a computer program that could do it fast, do it reliably, and do it for any protein you threw at it. And this brings us to the first massive breakthrough in the story. Back in the 90s, David Baker and his team of researchers decided, you know what, we're going to build that dream tool. And they called it the Rosetta Project. The whole entire project was built on one simple, beautiful idea from physics. And the idea is this. Out of all the zillions of ways a protein could possibly fold itself, nature always finds a way for it to settle into the one single shape that is the most stable, the one that has the lowest possible energy. Now, here's where it gets really wild. Rosetta doesn't just predict structures that already exist, it designs totally new ones. So let's say we want to create a brand new protein that's never existed before. We give Rosetta the basic backbone shape we're aiming for. Then the software just goes to town, cycling through millions and millions of amino acid combinations, calculating the energy level for every single one. It's relentlessly searching for that one magic sequence that creates the lowest energy, the most stable version of the shape we want. And once it finds it, we can actually synthesize the gene for it, pop it into some bacteria, and have them produce our custom-designed protein for us. It is literally bioengineering from the ground up. But Baker's team wasn't the only one in this race. While Rosetta was coming at the problem from the world of physics, another group was taking a completely different, but just as powerful approach. And their secret weapon was the rise of artificial intelligence. Right? So over at DeepMind, you had this team led by John Jumper and Demis Hassabis, and they were building a system they called AlphaFold. Their big idea wasn't just to use physics principles. No, they were going to teach a machine how to think like a biologist. AlphaFold's whole strategy was to learn from everything we as humans already knew. They fed the AI a gigantic database of every known protein structure. It just soaked it all up, learning to recognize patterns, to see how proteins actually fold in the real world. So it predicts the simple local structures first, and then it figures out the final 3D shape that plays by all the rules of biology. Burying the parts that hate water, exposing the parts that love it, and of course, finding that super stable, low energy state. So what you have here are two brilliant teams attacking the exact same problem from two totally different angles. One is working from the first principles of physics, the other is using data-driven AI. And guess what? Both of them achieved unbelievable success. And the result of all that? A Nobel Prize and the beginning of a whole new era in science and medicine. And so, for all their combined work, David Baker, John Jumper, and Demis Hassabis shared the 2024 Nobel Prize in Chemistry. It was this huge victory for both computational biology and artificial intelligence, and really a testament to how different paths can lead to the same incredible destination. And this is why this matters so much. This isn't just some abstract academic exercise. The ability to design proteins from scratch just blows the doors open to a whole new world of possibilities. We are talking about creating super-specific drugs that only hit their target, designing the next generation of vaccines, building enzymes that can literally eat plastic pollution, developing brand new molecular materials. I mean, the list just goes on and on. We finally have the tools to be architects of biology itself. But the story doesn't just end in some high-tech lab or with a Nobel Prize ceremony in Stockholm. See, David Baker has this really powerful belief that even the most complex science should be accessible to everyone, because you just never know where the next big breakthrough is going to come from. I want you to really think about this for a second. We usually picture science as this thing done by experts in white lab coats. But what if your brain, what if your intuition could help solve a puzzle that has had those same experts stumped for years? To put this incredible idea into practice, Baker's team helped create a video game called Fold It. And it's not just a game, it's a crowdsourcing platform for actual scientific discovery. It turns protein folding into a puzzle, and it lets anyone, you, me, anyone, use their spatial reasoning skills to try and find the lowest energy shape for a real-life protein. And the result of this experiment? Well, in one of the most incredible stories of citizen science ever, players of Fold It, gamers, managed to figure out the precise 3D structure of a critical enzyme from the HIV virus. This was a problem that professional scientists had been stuck on for over a decade, and gamers solved it. Just let that sink in for a minute. Which leaves us with one final massive question. We've gone from not even knowing how proteins fold to being able to design them from the ground up, even using video games to help us do it. We are now officially architects of the molecular world. So the only real question left is, what on earth should we build next?